Well, thanks very much, Marcus, for the invitation. I don't really understand very much because I'm not a mathematician, but I will try to tell you a little bit about the application. We have worked together in this project with Vedat and with Marcus. So we are trying to find an analgesic compound that is pH sensitive, and that is why the title is called the pH sensitive ligand. So the reason we do this is because we are interested in pain treatment, and you know, all of you have pain, headaches sometimes, or other types of pain, and we know that there are basically two different types of drugs. One type is the opioid class of drugs. They are usually called the strong painkillers, and then there are a second class of drugs, the so-called non-steroidal analgesics. So this is like an aspirin or diclofenac or ibuprofen. You have all heard of these names. The problem with all these drugs is that they have side effects, and especially the opioids have very serious side effects like addiction and respiratory arrest and sedation and nausea. So we have been working for a long time to find out whether we can make opioids that do not have those side effects. And the way we approach this problem is we know where the side effects are produced. They are produced in the brain. Most of these side effects, they affect the respiratory center in the brain. They affect the so-called vomiting center in the brain, and they cause sedation. All of this happens in the brain. And of course, the analgesic effects are also mediated in the brain, but luckily they are not only mediated in the brain, they're also mediated by peripheral opioid receptors on primary sensory neurons in the peripheral tissue. That's why this is shown down here. So opioid receptors are located in all three parts of the nervous system, in the brain, in the spinal cord, and in the peripheral nervous system. So we're trying to make a ligand that only works in periphery, in the peripheral tissues. So what we've done, and we've done this in the past by a very simple method, by just injecting small doses of morphine. Morphine is a prototype of an opioid agonist directly into peripheral tissue, like a knee joint, for example. And we know that this works very well. We have measured pain after knee joint operations. Pain is measured with a so-called visual analog scale, and we see that different doses of this morphine, 2-4 milligrams, cause dose-dependent analgesic effects compared to saline. This is the so-called placebo. So this works, and of course what we would like to do is we would like to design a drug that does not have to be directly injected into peripheral tissue, but it can be taken systemically, for example by the oral route or the intravenous route, but it only works in the periphery. So the next thing was we tried to figure out how much is actually mediated by peripheral opioid receptors when we inject a regular opioid intravenously. So we did a study in two separate hospitals in patients that underwent knee surgery, and we injected all of these patients received morphine after the operation by intravenous injection, but half of these patients received an antagonist that does not work in the brain. It only blocks peripheral opioid receptors outside of the brain. So the theory, the hypothesis was that when we block these receptors with this drug, which is called methyl naltrexone, then the patients will need more morphine because a part of the receptors outside of the brain is blocked. So this is what we actually found when we compare these different groups. These are the two different hospitals, number 1 and number 2, and we compare the group that receives the saline into the knee joint, that's the placebo group, to the other group that receives the antagonist into the knee joint, that was a systemic injection, we found exactly what we expected, that this group needs a lot more morphine after the operation than the placebo group. And we see that this effect is about almost 50% more morphine. So a big portion of morphine, the regular morphine analgesic effect is mediated by peripheral opioid receptors. So what we did in this project, together with Markus Weber's group and Vedat, we tried to exploit the fact that when we talk about peripheral injured tissue, there is a low pH. This has been known for a very, very long time. Many people have measured this back to about 100 years ago. We know when there is injured tissue, like after surgery, the pH is low. And the pH is low because there are a lot of cells that die, and that causes acidosis in the injured tissue. As opposed to the brain, or we also know there are receptors in the gut, where we have a normal pH, 7.4. So we try to exploit the fact that this difference of pH exists between 5 to 7 and 7.4, and the hypothesis was that the interaction between the ligand and the receptor is different at low pH compared to normal pH. And this is where Vedat's part comes in, where he simulated the two conditions in the computer. And he showed, and this is actually, there are other people that were involved. This is actually a very early pilot study done by Olga Sharkoy in Markus Weber's group. They showed that the ligand binds much stronger to the receptor. This is what you just saw in the last talk. The gray thing is the opioid receptor, and this is the ligand in low pH conditions compared to normal pH conditions. So there is a much bigger movement between the receptor and the ligand. The binding is much more unstable under pH 7.4 as compared to low pH. This is 5.5 here. So this was one of the initial findings that we found in these simulations, and the next step was then to create, to design a ligand that's shown up here that has a comparatively low pKa value. And the theory was that this ligand that has a pKa value that's actually much closer to the inflamed tissue, to the injured tissue, to the acidosis, that this ligand would only bind to the receptor under these conditions because the tertiary amine group here is only protonated under those conditions and not under normal pH conditions. And the reason why this is so important is because we have known this from many publications before, that this tertiary amine group is very important to bind to the receptor. This is the 7-transmembrane structure of this receptor shown here. And we know there are several amino acid groups that are very important. And especially this group here is very important for the binding between this tertiary amine and the amino acids in the receptor. So the way this was achieved, and this was done again in Markus Weber's group, is they simulated several different variations of this molecule, and the way they tried to achieve this lowering of the pKa was by adding fluorine atoms to this molecule. And fluorine atoms are known to be very electronegative, and therefore they pull away electrons from this tertiary amine group, and that means the tertiary amine group will only be protonated and able to bind to the receptor when there are many more protons in the environment, which means we need an acidic environment. So that was the theory, and they actually constructed this molecule in the computer. They've constructed different molecules. Here are two more variants shown underneath. Here you see that the fluorine atom is at different positions here. But we actually found out that this position is the best. It does what we want. It lowers the pKa to about 6.8. So we went to a chemical company in Berlin. They synthesized this compound according to the computer design, and then we tested this in an animal model. And this model has been used for a very long time in our group. It is a rat model where we compare an inflamed hind paw to a normal hind paw. Here you see the difference, and we apply pressure to this hind paw, and we measure the force at which the animal withdraws the paw, because it's painful. So when this threshold, you can measure this in newtons. When this goes up, it means the animal becomes less susceptible to this pressure stimulus, and therefore we find an analgesic effect. So here is what we found with this new molecule. We called this new molecule NFEP, N-F-E-P-P, NFEP. And we injected this intravenously, for example, in many different experiments. And we measured the paw pressure threshold in the inflamed paw compared to the non-inflamed paw. So this is what I just explained. We see that there is an increase in the paw pressure threshold when we inject this compound. And we compared this to regular fentanyl that does not have the fluorine, which actually has a very high pKa. The pKa of regular fentanyl is above 8, and therefore it's protonated both under normal and inflamed conditions, as opposed to the NFEP, which is only protonated under inflamed conditions. So what you see here is the effect of fentanyl. This is on the left side, you see this. This effect is after fentanyl, and this is after NFEP. So you see a bigger effect after fentanyl compared to NFEP. And the way we explain this is because fentanyl works both in the brain and outside of the brain, whereas this molecule, NFEP, only works outside of the brain. So the effect is a little bit smaller. But we tested for example, we tested also with a heat stimulus here. When we use heat instead of pressure onto the paw, we've actually found that both of these molecules are equally strong. Importantly, we then used an antagonist that only works outside of the brain to find out what is the portion of the analgesic effect that is mediated outside of the brain compared to the placebo group. So we did pretty much the same thing that we did in the human study just before, and we found out that the fentanyl effect, for example, here, is only antagonized by 50% by this molecule outside the brain. So half of the effect is mediated in the brain and half of the effect is mediated outside of the brain, whereas this effect by the NFEP, by the new molecule, is completely antagonized by this peripherally selective antagonist. So almost 100% of that effect is mediated outside of the brain. That was what we have hypothesized in the beginning. But more importantly, we wanted to see that there are no central side effects. That was the purpose of the whole idea. So we measured different central side effects. For example, one of them is called the addiction potential. We measure this with a test that's called place preference conditioning. It works by putting the rat several days into one box that has a specific environment, for example, a white wall, and then put it into another box that has a black wall, and you can pair the two environments either with a drug or with placebo. And on the 6th day, you do this for 6 or 5 days, and on the 6th day you put the rat in the middle. If the rat goes to the white box, it means it liked the drug. If it goes to the black box, it means it didn't like the drug. That's what we call condition place preference. So when this goes up, it means that there is a lot of drug liking. The drug worked in the brain. It caused what we call reward or addiction potential, drug liking. But when you don't see this on the right side, this is what we found with this new compound, NFEP. We found basically there is a dose. There is no effect statistically, and at higher doses, the effect becomes less. So this is a very good sign that it doesn't cause reward, it doesn't cause drug liking. We used other tests, for example, to measure sedation. This is what we call this. For example, the Rotor Rod Test, we put the rat on a rotating rod, and you measure how long the rat can stay on this rotating rod before they fall off. And this basically tells you something about motor coordination or sedation. So you can see that, again, fentanyl immediately causes this sedation effect because it works centrally. But the new drug, the NFEP, causes practically no effect in this test. And the same thing here. This is what we call the open field test. We basically measure how long the distance is that the rat travels in a set amount of time. You see there's a clear sedative effect that when you give fentanyl, which is a centrally acting opioid, as opposed to the new drug that has practically no effect. And the most important side effect is shown here is respiratory depression. We measure the oxygen saturation in the blood by a simple pulse oximeter that is used in patients every day in the operating room. And you see when you compare the new drug with saline with a placebo, there is practically no effect at all in oxygen saturation. So that's a very good sign that this causes no respiratory depression, no inhibition of breathing, and no inhibition of oxygen saturation. So what we've done in summary, we've managed by the help of this computer method that was explained by Vedat just before, that we actually found a drug that only works in inflamed tissue where you have a lot of protons, acidic tissue, acidosis, because only in this tissue the drug is protonated and can activate opioid receptors, versus in the brain or in the gut where there is normal pH, a smaller amount concentration of protons, the drug does not bind to the receptors and causes no central side effects. And that's really wonderful. And of course, we were very happy to find these effects, at least in rats. And we're now trying to develop this, hopefully at some point, for human use. But there is, of course, a very long way before we can actually test this in human patients. So these are all the colleagues that contributed to this project. Thank you very much.
Thank you very much.